So Dr. Azam, go ahead.
Sure, thank you, Jordan. And greetings from the heart to each and every one of you around the world who are attending the third webinar of this series of dialogues organized by the Radiation Research Society. Today, it is my privilege and honor to introduce Dr. Olga Kovalchuk, a member of our society, who will discuss with us the topic of sex and gender science in radiation research. Dr. Kovalchuk is professor and Board of Governors Research Chair at the University of Lethbridge in Alberta, Canada. She is one of a select few to hold a Canadian Institutes of Health Research Chair in gender and health. Through her career, Olga built a strong and internationally recognized program in epigenetics, cancer, and radiation research, with an emphasis on sex and gender to the epigenetics of health and disease, in particular to radiation biology. She is truly the catalyst that propelled this aspect in basic radiobiology. Dr. Kovalchuk holds many honors. She is one of 2 Canadians who are funded by the International Pediatric Oncology Experimental Therapeutic Investigators Consortium for her valuable expertise in epigenomics, sex and gender analysis, as well as precision medicine. Her laboratory was the first to apply the sex and gender lens to radiation research and has made several important discoveries that have the potential to shape our understanding of radiation health effects and other harmful exposures. Olga is a recipient of numerous awards, and today I want to recognize 2 specific awards, the YWCA Women of Distinction Award and Canada's Top 25 Women of Influence Awards, which she received twice. Most importantly, Olga trained numerous young investigators in the different aspects of the radiation sciences. These investigators now have thriving careers in various corners of the world and have earned numerous awards of their own, including the Michael Fry Award of the Radiation Research Society. Olga is truly a role model for our scholars in training and our early career investigators, in particular, our female scholars. And Olga, we thank you for taking the lead in this webinar, and we look forward to hearing you. Thank you very much. And a big thank you to Jordan and the team at AMBD, Katie, Veronica, and Audrey. Thanks, Olga. The screen is all yours, Olga.
Thank you very much for a very, very kind introduction. Can you hear me?
Yes, ma'am, you're good to go.
Hello? Excellent. So again, thank you so very much for this very, very kind introduction, Ed. And thank you to the Radiation Research Society for inviting me to give this webinar. And greetings to everybody who is listening to us. And thank you for everybody for joining. Sorry for the tech difficulties. You know, we have sometimes, you know, when you fully rely on internet, it's there for you, but sometimes it has a teeny bit of a glitch. But anyway, now we are set to go full speed ahead. So today, the webinar, the focus of the webinar is specifically sex and gender science in radiation research. And when I was thinking, if I could maybe even narrow it down or maybe broaden it up. So I decided, how about we put even another title to it and call it differences in radiation effects, and how we can go from personalized science to personalized protection. Can we? Should we? So let's think about that. So what do we mean when we talk about health? We wish each other good health these days. That's a very common thing. We used to do that always, but now we do that almost every day. So, but what is health? Health is a state of complete physical, social, and mental well-being. It's not just an absence of disease or infirmity. In the other words, when are you healthy? Not when nothing hurts, but when you are feeling great. And that definition hasn't been amended since 1948 as it was originally proposed by WHO. It was adopted at the International Health Conference. So it still stands, that very same definition. What influences our health? Pretty much our environment, because we exist in a very, very complex environment. So as this very, very elegant recent paper that came out on very interesting exposomes talks about an individual, a human being here, either male or female, being exposed to a variety of things, life around us, lifestyle, ecosystems, social environment, as well as physical and chemical environment. They all influence us and they all affect our health, either positively or negatively. And what we, of course, want to do is make sure that we minimize any potential harm and we maximize the potential benefit, and essentially we keep ourselves as healthy as possible for as long as it's possible. All right? So, but within the physical and chemical circle there, which is on the bottom, you can see what are those various types of chemicals that are around us, from various types of medications to various types of chemicals we use in a day-to-day basis, to some environmental exposures, occupational exposures, biological agents, viruses, well, these days very important, bacteria and other things. And of course, ionizing radiation. It's also one of the important exposure factors that exist around us, and it influences us. And we need it. We are exposed to environmental radiation. We are also exposed to man-made sources. So in terms of sources of radiation exposure, I think Radiation Research Society audience, of course, most certainly knows, but for the broader audience, which we really hope to bring in and get interested in our field, well, where does the radiation come from? About 82% comes from natural background radiation, and about 18% is man-made radiation. And if we were to break down that 18%, take it as 100 again, so where do those exposures come from? Diagnostic tests, as well as nuclear medicine, quite a big, kind of almost like a lion's share. There are some occupational exposures. There are most certainly fallouts that unfortunately have happened, as for example, Chernobyl disaster, Fukushima accident that has happened. There's some radioisotopes in consumer products. So, but overall, again, medical exposures, medical x-rays, like diagnostic and treatment exposures, again, they comprise the largest part of different types of exposures. So again, cosmic rays, building materials, water, food, our Earth are actually containing radioactive materials around us. The very interesting factor here, specifically for the province of Alberta, where I live right now, is radon. It's in the air, and it's actually quite, unfortunately or unfortunately, quite frequently. We get to it as a problem right now. Air travel, again, another source of radiation exposure. Plus, we also have most certainly nuclear power plants. We do have radioactive waste. Testing fallouts, unfortunately, happened before. Fortunately, it's not happening to that extent right now. But again, we do have a fair amount of diagnostic and therapeutic radiation that still comprise the lion's share of our exposures as human beings, like where are we exposed. And if we're talking about diagnostic and therapeutic procedures, more than 2 billion procedures are occurring worldwide per year. More than 2 billion procedures. So a lot of individuals are exposed to radiation. So we want to maximize the benefit and we want to minimize the risk, which is absolutely understandable. And as I mentioned, very interesting and very severe problem in Alberta is radon, naturally occurring gas. It's produced from radioactive decay of uranium that's in rocks and soil. It's gas, it's colorless, it's tasteless, it's odorless. You don't know it's there, but it's there. And it's quite interesting. It's very prevalent in Canadian prairies. If you were to look at the radon map of Canada, you can see the circle, the city of Calgary. That's where actually I now am, and this is where this webinar is broadcasted from my side. And as you can see, everything is actually red. So relative radon hazard is quite high in the province, in the prairie provinces. And if you were to look further, where why is that a problem? Radon, again, uranium decay to radium and further to radon, it's a gas. It leaks and it penetrates the house. It penetrates the foundation. So some of our colleagues at the University of Calgary have actually started a research project looking at the health effects of radon and trying to advocate for the need to actually radon-proof your homes to make sure that that unnecessary type of exposure, truly unnecessary, is not occurring. Why? Because if you were to look at the areas in the city of Calgary, there are some areas where actually the levels of radon are quite high. Reaching at the maximum level, almost 30 to 100 becquerel per cubic meter, which is actually quite a substantial contribution to the yearly dose. And therefore, of course, the effects of radon have to be studied and have to be studied in such a way, again, that we protect all individuals, men and women, boys and girls across the age continuum. And I really would like to play this little video. This little video is an excellent educational resource that was created by Institute of Gender and Health, which is one of the Institutes of Canadian Health Research.
So Dr. Kovalchuk, I sent everyone a link to that video. It was a little hard to hear, so everyone has a link to that video in the chat box.
Mm-hmm. Oh, okay. I would be really happy to then share that video and share the presentation. And because that's essentially the video, and why I really wanted to bring it up is how important it is to integrate sex and gender research in essentially pretty much everything we do. And I will then give the examples of how we went about integrating specifically sex as a biological variable, and later on how we looking to integrate gender as another variable, and more of a social cultural variable in our research. First, let's go and talk about definition. So when you talk about sex and gender, and I have reviewed many papers, and oftentimes I would comment when people will say, we use rats of both genders. Well, rats cannot be of both genders. A rat can have different sex, male or female. Gender refers to socially constructed roles that shape behaviors, activities, expectations, opportunities. Essentially refers to relationships between people and distribution of power in these relationships. So gender interacts in certain distinct forms with categories of biological sex. But what's really important to understand that, again, gender is essentially a psychosocial term, if you wish. It's a behavioral term, whereas sex refers to a biological term, to our X and Y chromosomes. Why gender is important? It's a very important determinant in health inequities. Separately, as well as if we're talking about intersections of socioeconomic status, bring in the ethnicity, disability, sexual orientation, and so on. So gender may influence employment, may influence working conditions, may influence career trajectories, may influence health and specifically for health and social care workers. So this all was identified and actually, stipulated by the World Health Organization. So again, in terms of sex and gender, sex is a biological distinction between males and females. Gender is a social difference between males and females. So, in this case, I will give you some of the examples of our work that we have done, starting first to analyze how males and females respond to radiation, whether or not they respond to radiation differently. It all started in 2003, with first publication coming in 2004, which were based on serendipitous discovery. And we wanted to understand the differences in between different tissue types. How would the global gene expression be affected by radiation in different tissue types? And at that point, we thought it would be prudent to use several litters of mice just to reduce the genetic noise and take a look at how the same dose applied but received by different organs will be affecting the gene expression pattern. And to our huge surprise, what we discovered that actually the transcriptome response, the response to acute and chronic low-dose radiation was very, very dissimilar in male and female mice. And this was absolutely, a serendipitous discovery that we made. So what we did there, when we say chronic, it's more of a fractionated type of exposure where animals were irradiated once every 10 days, and what we call acute dose was a lump sum dose, which was actually a sum of all the doses were deposited over 10 days. And then we just compared the levels of the gene expression patterns in these animals, and we looked in different tissue types. So what we found was that actually differences between males and females were actually very, very striking. AF stands for acutely exposed female, single dose. CHF stands for chronic, and then again M stands for male. And you can see in this table, you can see for yourself that actually the differences are quite striking. So there are very pronounced differences in many different genes that are regulating DNA repair, that are regulating overall genome, responsible for genome stability, that are regulating development, and so on and so forth, as well as inflammation. So when you looked at some of the receptor coding genes, similarly, we found numerous differences in various receptor coding genes between males and females. So this was actually essentially the first work that we've done that started the whole series of works in this field, where we decided to understand why, first off, whether it's indeed true, and why is it happening, and what's going on, and how significant is it? So we also looked at DNA repair genes, and again, we had seen quite significant difference in the expression even of DNA repair genes. Something that you think should be so fundamental, should be pretty much even across different organisms, right? But well, not necessarily. In some cases, some of the expression of some genes goes up in males and down in females. Or sometimes the deregulation is way more pronounced, for example, in males than it is in females. So then we proceeded and we started looking at different regions and different regions at different tissue types and different regions of the brain. And what we also found that brain responses to also acute and chronic low-dose radiation were actually also sex-specific. There were very significant differences between males and females. And there, what we have done, we have used similar type of exposure again, chronically or projected exposure, fractionated exposures, we call it chronic, would be animals receiving exposures for once every 10 days, and the acute dose receives the lump sum dose for over just once. Then animals were sacrificed, and gene expression levels were analyzed. And what we found, again, when we looked at hippocampus and prefrontal cortex of males and females, there were very, very significant differences between male and female responses. And in order to understand the nature and the mechanism behind these sex differences, what we decided to do is we conducted the experiments. We gonadectomized animals, and we tried to see whether or not in overectomized animals, we will see any differences as compared to females, and whether or not supplementation with estrogen will also reverse or at least partially reverse these changes. And yes, we have seen that oftentimes in animals that were overoptimized, the patterns have changed. So which means that hormones do play a role in modulating transcriptional responses to radiation. Right now, when we know so much more than we knew pretty much 15 years ago, we understand that that's exactly the case. But this was essentially the start of our trip. Then when we looked at whether or not the molecular changes we have seen would manifest on the cellular level, on the level of neuroanatomical changes, we looked at hippocampal neurogenesis and whether or not radiation exposure will yield differences in hippocampal neurogenesis in males and females. And yes, absolutely. And you can see, again, males on the panel A and the ones below, and females panel B and the ones below. And you can definitely see, first off, there are some differences there from the very beginning. And then you can see that definitely there are differences in the patterns of responses of what we were looking at, the newly formed neurons, that were stained by double cortin. So you definitely can see that there are definitely differences. And again, removal of gonads and in overoptimized cohorts, as well as hormone supplementation, did influence the pattern. So again, there is a huge contribution of hormones in the radiation response sort of network, if you wish. Well, we proceeded and we started looking at whether or not exposure to what we call bystander effects or in vivo bystander effects, oftentimes probably more correct to call them abscopal effects. So whereby we expose one part of the body and look for the effects of radiation in the distal parts that were not directly exposed, albeit, of course, they probably received certain types of scatter radiation and most certainly received some systemic communication from these radiated parts. But what's most important is we have seen that, again, both whole body exposure and cranial exposure, because in that experiment we exposed only the head of the animal, whereas the body was fully protected by the lead shield, led to very pronounced and sex-specific changes in DNA methylation as seen in urine spleen. So we exposed the head of the animals and we only looked at changes in spleen, which was reasonably far away from the field. Of course, it did receive certain type of scatter. But again, what's the most striking is the pattern. It's very different between males and females. And that specific pattern, quite interestingly, was altered when, again, we removed the gonads. So we overoptimized the animals or we castrated the animals. So, and there we have seen that as soon as you remove gonads, the responses, and specifically the responses to the direct radiation exposure to the essentially when the whole body got exposed, they're almost identical. So as soon as the hormones are out of question, out of the picture, responses become very similar. So which means we have hypothesized that yes, hormones do play a role. We also looked at other components of the molecular networking. We looked at small RNAs, microRNAs, for example, in this case. And what we also identified is that specifically in male and female mice that were subjected to whole body exposure or to head only exposure of long-wave X-ray, we have identified significant changes in microRNAs. But these changes, as you can see in the slide, were actually quite sex-specific, which was very, very interesting. There were some similarities, most certainly, but there were actually some differences that were also identified. So in terms of similarities, we identified some quite interesting microRNAs. So, for example, microRNA 24 and microRNA 23B, they were found to target Notch, very important receptor involved in cell-cell communication. And again, a microRNA 24 was shown to target P38, protein that was previously indicated by Bystander responses. And we have also noticed that actually there were indeed differences in the levels of notch, and there were actually sex differences in the levels, again, of these proteins. So again, there are sex differences. So males and females in the animal model of ours did not respond to radiation absolutely similarly. There definitely were quite significant differences. The differences, I want to say, were eradicated, but that's not true. They changed. The pattern has changed once we removed the code. In gonadectomized animals, the patterns have changed, which again makes sense because one could say, of course, small RNAs are regulated through hormonal signaling as well. And as soon as you have an interplay between regulation of radiation response and hormonal response, most certainly you are getting the different response on the level of, pretty much on the level of microRNA. We also found that the changes in the protein called diester, the one that actually produces or leads to maturation of microRNAs, was also affected by radiation exposure, but was also slightly sex-specific. There were definitely differences between males and females. There they were, I would say, less pronounced, but they also, they essentially also occurred. And again, the role of gonads also played certain part in the, essentially in their, in the levels of those proteins. If you're interested, there are some references to the articles we have published, and so we have done some research, and later on we proceeded looking into bystander effects in other tissues, because in the initial experiments that were conducted by very, very talented colleagues and students in my lab, starting my lab, Dr. Kovalchuk. So in the series of experiments we started, we have used bystander models whereby we would expose the brain and look at the effects in the other somatic tissues. Then we thought, we need to do the opposite. We need to see whether or not the brain is affected and whether or not actually those changes again are sex specific. Why would we do that? Because we are very interested in the effects of radiation therapy. Radiation therapies are very common, and oftentimes people are complaining after radiation therapy about cognitive issues, memory loss, so on and so forth. So again, bystander effects, as I mentioned, have been studied in cell cultures and tissue explants. Dr. Azam has done numerous studies and essentially laid the foundation for the whole field. And what we decided to do, we decided to look at whether or not radiation therapy-like exposures of somatic organs, for example, liver, will cause bystander effects in the shielded brain, and whether or not these effects would be the same or would they be different in males and females. This was our experimental setup. Again, we had males and females in each group. Animals were heavily exposed, and animals that, and those were rats, those were LE rats, whereby we shielded the entire body and only had a small portion open so that we irradiate the liver. So dose to the brain was measured, and I published our measurements in, I think, in the International Journal of Radiation Research. So it was 24.6 centigrade to the brain of the animal when it was, when the entire animal was irradiated, whereas when only the liver was irradiated, brain received 0.125 centigrade as essentially the scatter. And then what we have done, 14 days post-exposure, we have done molecular analysis, we've done behavioral studies, and neuroanatomical studies. In terms of molecular analysis, the first thing we did, we have done global transcriptome profiling using RNA-seq. We looked at hippocampus and prefrontal cortex, two key areas involved in memory, cognition, as well as essentially a variety of really important behaviors. So, in what we were completely stunned to see that, first off, in prefrontal cortex of females, changes were way more pronounced than males. And somehow these specific dose, and here we profiled only sham-irradiated versus liver-irradiated animals. So changes were not that dramatic, albeit differentially expressed genes in females were involved in blood-brain barrier, were involved in apoptosis and were involved in neuroinflammation. So they were actually relevant to what was happening to the animal. When we looked at neuroanatomy, and we have done the Kovalchuk staining, and we started looking at the central neuronal morphology by looking at the spines, the branches, and the length of the branches in different regions, in parietal cortex, medial prefrontal cortex, or orbital frontal cortex and hippocampus. We were absolutely surprised to see stunning differences between males and females. It definitely looks like, first, both head and liver radiation affect neuronal morphology, and females are definitely more affected. When we started looking at behavioral outcomes, and we have tested the animals for the open field activity, elevated plus maze, and novel object recognition, these tests are sensitive to hippocampal and prefrontal cortex injury. So what we have noticed that, again, there were definitely sex differences in behavioral patterns. And again, it looks like females are more effective than males. So this was very interesting for us. And we decided to follow because we really wanted to understand what is the contribution of scatter radiation to this. And we designed another experiment where we wanted to exclude transfer of any signals via blood. So to do so, we exposed one animal. The animal that was exposed was there, and the animal's liver was exposed. And the other animal was positioned nearby, fully shielded in such a way that its head, its brain, was on pretty much almost the same distance from the brain coming out of the exposed animal. And we essentially looked at that scatter exposed animal, just to model scatter exposures. We measured the scatter dose to that brain. It was 0.115 centigrade. And when we looked again at global gene expression, we were really blown away to see the differences between males and females. Again, way more changes were seen in females than in males. And when we looked at downregulated genes, they belong to neuroactive lichen signaling, to calcium signaling, axon guidance, neurotrophin signaling pathways, all of those pathways that actually are those, I would say, the foundation for the proper brain function, if you wish. So just look at this axon guidance pathway in the brain of exposed females, whereby green is essentially showing the downregulation, red is kind of, from -1 to +1, and each of these kind of little lines, I hope you can see them. It's a single animal, so she's red leaf. I think so. So it's pretty much the entire pathway is almost downregulated, which is really very important because those are the molecular changes that could then explain why indeed are we seeing changes in biology. And we did start looking at changes in neuroanatomy. And yes, lo and behold, we did see very significant changes in females. Specifically, again, females were more affected. And the level of, they had less spines were less long, and there was less branching in the female animals that were scatter exposed. Out of the behavioral tests, we also noticed that there were several differences in the outcomes of the elevated plasmase. Pardon my pardon the typo here. So that is essentially the test that looks for anxiety-like behaviors. So again, there are definitely such differences on the levels of molecular levels, on neuroanatomical cellular levels, as well as on behavioral levels between males and females, as in terms of how they respond to low-dose radiation to the brain. So again, we have done quite a lot of analysis then on, they were summarized in the previous, essentially in the previous table, but these are some of the studies that we have done looking again at each individual specific parameter, how would scatter radiation affect spine density, and you can see definitely sex differences there. How does it affect dendritic branches? Again, you can see definitely sex differences, specifically in C2-3 apical branches and C2-3 basilar branches. And overall, we concluded that both bystander and what we call scatter exposure, first off, they do affect the brain. They affect behavior and affect sex-specific. So if we're talking about liver radiation, it causes DNA damage, changes in gene expression, changes in morphology in the brain, affects behavior. And scatter radiation, essentially when just the scatter went into that, essentially hit that brain from the radiation being delivered to the nearby animal without any blood circulation. Again, we see altered gene expression, alterations in morphology, and affected behavior. So conclusion number one was actually brain is sensitive to radiation. Contrary to some former beliefs that brain is one of the radiation insensitive kind of organs. Why? Because we were always thinking about radiation affecting tissues that are predominantly highly, high replicated potential, if you wish, that are replicated at high rates, whereas neurons are not. But actually, that is not the case. Brain is very, very sensitive. So if we look at sex differences in response to ionizing radiation, overall, I just gave you a couple of examples. A lot of studies have been done, but we have also looked at trying to summarize what's known about responses in humans. And we recently had a review article published in Frontiers where we summarize sex differences in response to ionizing radiation. Looking at, for example, cohorts in Chernobyl nuclear reactor incident. So differences, overall differences in cancer rates, and as well as differences in, for example, in rates in childhood cancer rates in males, for example, when mainly due to leukemia. There are definitely sex differences in the analysis that were identified looking at ABOM survivors. There are definitely differences in occupational exposure cohorts. Because males, for example, have increased relative risk of cancer of colon, pancreas, and testes, whereas in females, the primarily risk is infertility, and then there is a relatively high risk for grade for lung cancer, liver cancer, and bone cancer. So the patterns of radiation-induced cancers in males and females, to some degree, are different, and we really need to start trying to understand why are they different in order to make sure that everybody is equally protected. I'll give you one tiny glimpse of how we further continue these studies, because while we were interested in looking in radiation effects on the brain, we also wanted to see whether chemotherapy exposures are having any sex-specific effect. And why? Again, because there is a phenomenon we call cancer-related cognitive impairment, or chemobrain, where people pretty much survival up to 3/4 of people with cancer experience cognitive problems. They affect their lives on every level. And unfortunately, I had the chance to see for myself as to how difficult post-radiation, post-chemotherapy effects can be on a person. So here we had, again, an mouse model where we exposed mice to mitomycin C cyclophosphamide with saline, pretty big groups of animals we had there, and we looked at gene expression, and we looked at prefrontal cortex in animals exposed to MMC here. And you can definitely see pretty much the figure speaks for itself. It's very, very sex-specific. There are definitely differences between males and females. If you even look at oxidative DNA damage, there are differences in males and females. You could say they're not that drastic, but there are some differences between males and females in the levels of ACKNOWLEDGEMENT genes. And there are some differences between males and females in the levels of 5-methyl C and 5-hydroxymethyl C. Again, there definitely are differences in females, but they're not statistically significant in males. So when we looked at it overall, we found that chemotherapy, specifically cytotoxic chemo, I couldn't call mitomycin C, I don't think it's correct to call it radiomimetic because it's kind of not. But again, it's a cytotoxic chemo. What it does, to a certain degree, similar to radiation, it leads to accumulation and persistence of oxidative stress and DNA damage, altered methylation, aberrant gene expression that are sex-specific. And where would I want to, if I were to wrap it up, where do I want to take it? And what type of take-home message do I want to propose for, this webinar, which we just scratched the surface of this issue of sex and gender differences? Recently, I started collaborating with Gender Plus Radiation Impact Project, or the GRIP, and the wonderful person who organizes, Mary Olson, has published a very interesting article, Disproportionate Impact of Radiation and Radiation Isolation. And there, as you can see on this slide, it's very telling. You can see the increased risk by age at exposure to 20 millisieverts of radiation. So you can see that if we look at males and at females, and the risk is plotted as age at time exposure on the x-axis and cases per 100,000 age peers on the y-axis, what you can definitely see that the risks of developing cancer are much higher in females than they are in males. Our radiation protection guidelines are based on reference male. Manhattan Project participants are reference male, around 30 years median age. So if we would compare the risk of a 30-year-old male with the risk of a 10-year-old female, we are talking about, I would say, over 5 times difference in the risk. So even if we compare males and females of the same age, 20, there's definitely a difference. 30, there's a difference. Pretty much a difference almost goes to, almost kind of disappears if we're talking about, I would say, 65 and older. Again, why? Probably because of sex hormones. But even though we don't fully understand 100% why, it's very important to understand that this is the case, that this exists, and we need to understand these risks, and we need to try to be able to work on them. And these were the questions that Mary posed in her article, and I found them very inspiring and very interesting, and we're trying to move that direction. Why is biological sex a factor in radiation harm? Why is sex difference in radiation harm greatest in young children? And that's actually true. Young girls, little girls, are the most susceptible to radiation exposure. Should we think about that when we prescribe CT scans to pediatric patients? Should we prescribe CT scans to pediatric patients always, or should we really consider in a way, we of course are, but sometimes, cases occur and parents ask, let's do a CT scan. Well, what type of harm are we doing there? Is percentage of reproductive tissue a factor due to greater reactivity of this tissue to radiation? Is percentage of fat tissue a factor? Essentially, what are the differences on cellular levels and biological differences? What are the data sets? Where can we get the data sets? How can we create the best possible data sets? Through now that we have AI capabilities, that we can mine the data, we can mine health records, trying to pinpoint the differences and trying to pinpoint the potential mechanisms of sex differences. And do we need to reevaluate and revisit the protection guidelines? Do we need to make them sex specific? How difficult it is? Is it even important? In my mind, it probably is, but those are very, very open questions. I do have another video, but for the sake of, and I really hope you can take a look, it's, again, one of the wonderful resources learning about sex and gender. Why is it important to include animals of both sexes in our studies? Why is it important to include males and females always in clinical trials? Why is it important to even have cell lines that are sourced from males and females, in order to make sure that we really do not lose the baby with the water, that we do not say, oh, there's no fact, whereas the fact actually would have been noticed if we would have included females. Because a couple of decades ago, people continued to do research, biological research, on just male rats. Why? Because it's so difficult to deal with those females, with the extra cycle. But now we understand that we ought to do this. So at this point, I really would essentially, to sum it up, first off, I definitely want to acknowledge so many people with whom I've been blessed to work. My group, and that would be probably a slide as big as this to list everybody, all of my current students, postdocs, alumni, some of them, as I mentioned, are members of Radiation Research Society, and I'm so really happy about it. My wonderful collaborators, University of Calgary, as well as University of Alberta, collaborators all over the globe, specifically my colleagues in in Australia, my colleagues in Sweden, colleagues in NCCR, Dr. Popovny, colleagues in Russia and Ukraine, and wonderful funding agencies who have contributed to this research. And one very, very important aspect that we are now starting moving forward, moving towards and kind of entering, is we absolutely need to understand the effects, radiation effects in transgender individuals. Because we need to make sure that everybody is equally protected, regardless of their sex and gender, regardless of their age. Everybody ought to be equally protected. And for that, we really need to do more research. Why? Because, as in one of the first slides I mentioned, sex and gender are one of the biggest, essentially, discrimination issues, if you wish. And in order to make sure that everybody is equally protected and everybody's health is equally valuable, we need to do the research in such a way that we include male and female models in animal research, in cell research, as well as in human research. And we are really open to collaboration. We are very, very happy to collaborate. And I think right now, even though there's a lot of research, essentially the wet labs is put on hold, I think we all could contribute to data mining. We're trying to put together several meta-analysis, and we would really welcome collaborations. If anybody who is listening would be interested to collaborate. Thank you so much for listening to me, and I wish you best health these days. Stay safe, and let's discuss if we have questions.
So yes, thank you, Dr. Kovalchuk. Dr. Azam, I'm going to put the questions up on the screen if you wouldn't mind reading those off for Dr. Kovalchuk.
Sure. I have a question here, Jordan, that considering, on the screen, I have one here. Considering that there are differences between male and female as far as methylation events in bystander cells are concerned, would you expect differences in expression of abscopal effects that are detected during radiotherapy, Olga?
Let's be frank about it. We haven't studied it, but based on if we are able and if we're allowed to extrapolate from the animal data that we see, because again, we have done analysis using mice and using rats, and we have in fact differences in both mice and rats, so in two different species. So one could suggest that based on this, we would hypothesize and predict that yes, those differences, there must be differences in abscopal effects in radiation therapy. And it would be very, very interesting to study, to form a cohort, for example, of individuals undergoing radiation therapy, or form a cohort of people undergoing, for example, radiation diagnostics, and say, okay, we would like to see the biomarkers before and after exposure to therapy, males and females. There could be, of course, a couple of candidates there because individuals going for radiation therapy and radiation diagnostics do have an underlying disease, right? So they do have an underlying disease. And the contribution of, oftentimes there are certain sex specificities in those disease manifestations. But still, I think that would be the first step to do it. I would think there would be, and really we need to, let's say if we actually want to do some meta-analysis to pull out what's available. Let's analyze everything, but as a function of sex and gender. Of sex is easy, of gender is not easy because nobody really, charted again, until recently, the gender identification of individuals.
Thank you, Olga. We have another question is, I guess the question, did the changes in gene expression between male and female affect the induction modulation of signaling pathways? I guess the question is coming biomarkers to adverse outcome pathways and this. So have you taken a bioinformatics study of your wealth of data that you have accumulated to look at signaling pathways?
Yes, the answer is yes, because when we analyze the global gene expression, the way we analyze them, we actually map the signal pathways, right? Because gene expression changes only make sense if essentially if you're looking at the entire pathway that has changed, right? It's like, you have the whole army attacking your one soldier. If it's just one gene, well, if it's the whole pathway, then you really think about, oh, that's a problem. So, and yes, we have seen some differences in the pathways in terms of the level of their activation or inactivation. And oftentimes, our hypothesis is because in some of those pathways, hormonal regulation also kicks in. And as soon as we have hormonal regulated transcriptional response, we could expect the difference, right? We could expect that interplay. And in terms of, we tried to play around a little bit with hormone supplementation, try to see whether supplementation, for example, with estrogen could change those pathways back. And yes, it does to a certain degree, but estrogen is not the only hormone that's removed when we remove gonads, right? It's more than just estrogen. So we'll need to do more reconstitution experiments, if you wish, bring in other hormones and see how they play.
Thank you. And we have another question. Do you have information on the effect of dose rate between radiation dose rate on changes in gene expression between male and female?
I think in our experiment, in our experiment, I believe we had the same dose rate. But right now, what we're trying to do is we're trying to mine more data to see what is also available in the literature. And I think if we try to accumulate all the data in the literature and actually, again, stratify it by sex and go with everything, dose rate, diseases, outcomes, exposure types, but bring in sex as a variable, that's where we'll answer that question, I would say fully.
Beautiful.
Thank you. I'm not sure, Jordan, if we have additional questions.
Actually, no, we don't have any more questions, but I think what the next step is going to be is to let everybody know that we are going to be pushing this out so that they have the option to review the slides as well as the presentation and all the external links that were provided. So other than that, I just want to thank Dr. Kovalchuk for coming on, as well as Dr. Azam, and we look forward to receiving that information and putting it out to all of our RS members.
Thank you, Olga, for a wonderful webinar. Thanks a lot on behalf of everyone. Thank you, and wishing you and everyone the very best during this time, wherever we are in the world. Thank you.
Thank you so much, Ed, for again your kind introduction, for being a moderator, and thank you, John, for invitation, and to again all the Radiation Research Society friends and colleagues for inviting me. So let's use this time now as a time to analyze our data and think about future collaborations forward. And I wish everybody all the best.
Thank you.